the Enzyme Lab is really neat because what we're going to be able to do is to experiment with a little bit of digestion. And most of the time when we eat food, we don't really think about what happens after we enjoy how delicious it tastes and feeling full. But what will happen is our pancreas will secrete enzymes, and those enzymes are going to then be in our small intestine, and they're going to start to break down our food. They're going to break down those great big molecules into smaller molecules, break the polymers into those monomers, those small units. Now remember, the smallest unit of a carbohydrate, a monomer for a carbohydrate, is called a monosaccharide. The monomer for a protein is called an amino acid. The monomer for a lipid is going to be the glycerol group and the fatty acid chain. So enzymes are going to do that work of breaking the big polymers down into individual monomers. Now, one of the things that we need to think about is what an enzyme is. An enzyme is a protein with a very specific shape. Remember, our enzymes generally only work on one, maybe two types of substrates. A substrate is just anything an enzyme is working on. If an enzyme is doing an anabolic reaction, it is making bonds. It is bringing things together. If an enzyme is doing a catabolic reaction, it is breaking those bonds, breaking a polymer, in a lot of cases, into individual monomers. Now, if the shape of a protein gets changed, we call that denaturing. Now, there are lots of things that can denature an enzyme. The changes in pH, either becoming too acidic or becoming too alkaline and more of a base. The use of heat is an excellent way to change or denature a protein, as is the use of cold to change the shape of a protein. So those are some of the things that we're going to experiment with today. Now, the enzyme that we're going to be using will be lactase. That ending ASE is often used to communicate that we have an enzyme. Now, lactase is an enzyme that some of our bodies make. Lactase will break apart the disaccharide lactose. Now, lactose is a sugar that is found in dairy products. It is two monosaccharides that have bonded together. And what the lactase enzyme can do is it can do that catabolic reaction. It can break the bonds and separate the disaccharide into individual monosaccharides. Now, if somebody has lactose intolerance, essentially what we're saying is that their body is not making the lactase enzyme. They can't break apart the disaccharide. And so that lactose sugar, that disaccharide, sits in their large intestine, and essentially it starts to feed the bacteria. And the bacteria will start to produce some gas, which makes their belly feel unsettled. And all of that solute, all of that lactose solute in their large intestine pulls water into the large intestine and gives them that other feeling of diarrhea. So having lactose intolerance is really, really problematic for a lot of people. So in today's experiment, it is absolutely crucial that the milk that you get is dairy milk. We want to make sure that that lactose sugar is in here so that when we are using the lactase enzyme, that we can break apart that lactose sugar. Now, as you have noticed, one of our rules is being violated here. Remember to try and keep all pets, children, and curious adults out of your workspace. Hopefully at this point they are getting used to you doing experiments and aren't interrupting you too frequently. So over here on this side where I have all of my hands-on lab kit supplies, the one thing that I did was I did break open the bag full of supplies, and you'll notice in the pictures that they've got all of these little containers sitting with the tip up. So go ahead and get that set up early on in your experiment, or keep in mind that this is one way to store those little containers. We're going to be using the well plate in order to hold our test tubes, so we don't need our test tube rack. And then don't forget to find these little glucose strips, and we'll talk about those in a little bit when we get into the experiment. So hopefully today's experiment is interesting and is applicable, and you understand how it applies to our digestive system. Familiar with how lactase works. Now, one of the things I noticed when I was making up my lactase solution and crushing my pill is that the pill really didn't crush as nicely as I would have liked. So I went ahead and dropped it in, and then I've just been vigorously mixing this to try and get a really homogeneous solution. And the same thing with my 5% sucrose solution is I want to make sure to get this really, really, really mixed up. So we have our our 4 solutions that are made, and then I grabbed the 5th solution, the glucose that is at 20%, out of my little baggie, because we're going to need that for experiment 1. Now for experiment 1, we're going to be using these wells, and I know that the little dimples, those little indentations, are going to be harder for me to read for A, B, C, D, and E, so I just grabbed a white paper towel and wrote A, B, C, D, and E. This is going to make it a little bit easier for me to keep track of when I am doing my experiment. The other thing that I did was I grabbed my little glucose strips and I labeled those A, B, C, D, and E. But make sure that you do not label on the green part. We're going to need the green part. That's the part we need to change color. So, into well A, we are going to put our 5% sucrose solution, 1, 2, 3 drops of the 5% sucrose, and we're going to put our 3 drops of distilled water. Into well B, we're going to put 3 drops of milk, and you'll notice that I just compulsively mix every time I grab a solution. I'll add to well B my 3 drops of distilled water. Into well C, I'm going to add my 3 drops of sucrose solution. 1, 2, 3. Then I'm going to add 3 drops of lactase. Into D, I'm going to add 3 drops of milk and 3 drops of lactase. And then into well E, I'm going to add my 3 drops of very concentrated or stronger concentration of glucose solution. And then I'm going to add my distilled water to well E. Now I want to give this just a little bit of a shake. Try to make sure that everything gets mixed up. The next thing that I'm going to do is let these sit for 5 minutes. After 5 minutes, I can then test with my strips to see whether or not I have any glucose. So I've let my wells and my liquids set for the 5 minutes. The next step is going to be to take each of my glucose strips and just lightly dip them into the well. We don't need them to get too overly saturated with liquids. The liquid only needs to remain on there 5 seconds, to start to get a color change. And what I have here is I have this little strip that I dipped in a very high concentration of glucose, and it immediately changed color. When I compared it to the color chart, and what you'll have to do is click on the hyperlink within the protocols to get the color chart. This is telling me that I had a very, very high concentration of glucose, but I already knew that. That's why I tested it. And then here, I tested a solution that I knew definitely did not have any glucose. And so it did not give me a response at all. It's still the same color as the paper originally. It's almost the same color as my gloves. It is giving me a negative response. So with experiment 1, what we can do with that color chart and these little glucose strips is we can start to get not only a feel of whether or not our solution still has any monosaccharides, has any glucose monosaccharides, but then we can also get a feel for how concentrated. So again, this is going to help us to really understand whether or not our lactase enzyme over here was doing any work on the lactose, if our lactase enzyme was able to break that lactose apart. So, Now that you've got, hopefully, a feel for how this enzyme works, you're ready to move into the next part of the experiment where we get to play around with this enzyme. We get to really test its boundaries. How far can we push it before it starts to denature, before it starts to change shape and it will no longer work for us? And that's when things get really interesting, is where we start to find those limits of where something like lactase can work and can function. As always, email me if you have any questions.